Good evening, everybody. Today, Henrik Nielsen and his colleague will speak on the DeepLoc package, which is the software package which can predict localization of certain proteins. And it might be quite useful for the ongoing CAFA challenge. So, Henrik, thank you very much that you agreed to make the talk, and please start.
Thank you. So I don't have very many slides today, so I hope we can make this very much into a question and answer session. But I will start by just saying a little bit about the problem itself, so that we are on the same page with what this is all about. So each compartment in a cell has its own characteristic set of proteins. And how does this, do these differences arise? How does the cell know where to put which proteins? It turns out there are sorting signals, sometimes described as zip codes in the amino acid sequence. And this was actually the discovery that earned Günter Blobel the Nobel Prize in 1999. And the sorting signals can be predicted from sequences by machine learning. In my group, we have constructed signal P that predicts signal peptides for secretion, and target P, which predicts transit peptides for import into mitochondria and chloroplasts, and DeepLoc, which I'm going to talk about today, that predicts eukaryotic subcellular location in 10 categories. Now, the overview, this is also the graphical abstract of the paper. So we have a query sequence. It's processed by a transformer language model that makes each position in the sequence into a high-dimensional vector. This is processed by an attention layer and a multi-layer perceptron that makes two predictions. One is for subcellular location, 10 different locations, and I'll return to what exactly those 10 locations are. And also sorting signals prediction. We predict the presence or absence of 9 different sorting signals. For instance, signal peptides or GPI anchors. The new features of DeepLoc compared to DeepLoc 1. That was first, we wished to utilize pre-trained language models for subcellular localization. And why do we want to use protein language models? That is mainly to take advantage of all the unlabeled sequence data which is there in the UniProt database. For instance, there are maybe only in the order of a few hundred annotated peroxisomal proteins with experimental evidence in UniProt, but there are thousands and thousands of existing peroxisomal proteins in the database that are just not annotated as such. So could the model take that into account? It can work by using pre-trained language models. And another advantage is we get rid of the time-consuming profile generation. The accurate mode of DeepLoc 1 used profiles for encoding sequences, and constructing these profiles via database search takes time and computer resources. Then the second new feature is that we want to predict multi-label localization. As you probably know, some proteins can have 2 or more localizations. This is particularly seen for cytoplasm and nucleus dual localization, where proteins can shuttle between the cytoplasm and the nucleus many times during their lifetime. And then we also wanted to integrate this sorting signal identification into the prediction model. So we had 2 sources of data. One was the SwissProt localization dataset, UniProt release 2021-03, which were filtered using the following criteria. They should be eukaryotic, not fragments, because fragments could have N-terminal or C-terminal sorting signals missing. They should be encoded in the nucleus, that is, we exclude proteins encoded in the mitochondria or plastids. At least 40 amino acids long and experimentally annotated subcellular localizations. We distinguished 10 locations: cytoplasm, nucleus, extracellular, cell membrane, mitochondrion, plastid, endoplasmic reticulum, lysosome, /vacuole, Golgi apparatus, and peroxisome. You might ask, why do we fuse lysosome and vacuole into one category? That is because in plants and fungi, the vacuole is actually homologous to the lysosome in animals. Then we also have the human protein atlas data, which is subcellular localization annotation of human proteins using confocal microscopy. And that is a source of annotation that is independent of SwissProt. We use only the enhanced and supported annotations, and we reduce the data set, so only those with, it should have been less than 30%, not more than 30%. Less than 30% global sequence identity with the SwissProt dataset. So the composition of the datasets look like this. As you can see, nucleus and cytoplasm are the 2 big categories, and then we have extracellular, mitochondria, and cell membrane, also quite large categories, while peroxisome is so small it's not even labeled. See if I can use the HPA dataset. There are some categories missing. Most importantly, the extracellular category is simply not there in the human protein atlas because of the way the data are made. And the lysosome vacuole and the peroxisome were so small that we decided to exclude those categories. And then, of course, plastid is not present in a human dataset. At the bottom, you can see how many proteins had multiple locations. There were in the SwissProt dataset, there were examples of proteins with up to 5 locations, but they were really rare. And in the HPA dataset, only proteins with up to 3 locations were found. So then we also had these annotated sorting signals. These are signal peptides, which we got mainly from Signal P dataset, transmembrane domains, and we only annotate the first transmembrane domain because there are some results in the literature that suggest that the first transmembrane domain is more important for localization or sorting of the protein than the other domains are. Then there are mitochondrial and chloroplast transit peptides, and the thylakoidal lumen composite transit peptides. That is a chloroplast transit peptide plus a signal peptide-like sequence that directs the protein to be imported into a chloroplast and further into the lumen of the thylakoid membrane. Then we have nuclear localization signals and nuclear export signals, and these peroxisome targeting signals, PTS, where we used the SwissProt motif annotations to contain either peroxisome, peroxisomal, or microbody in the description. The sources, this is just the sources for the preceding slide. I won't go through them in detail here, but I will say a little bit about how we partitioned the data. Instead of homology reduction, we partitioned the data set with a 4-step algorithm that ensures that no sequence in a partition has identity above the threshold to any sequence in any other partition. And the threshold in this case was 30% identity in a global pairwise alignment. So the algorithm also ensures that all partitions have approximately the same number of sequences for each label. And then in contrast to a homology reduction approach like the HOBOM algorithms, it keeps as many sequences as possible retained in the dataset overall. So for the training, we used a fixed language model. That is, the language models were not fine-tuned. We did experiment with fine-tuning, but in our case, it did not help. That is in contrast to Signal P6, where we saw that fine-tuning the language model did help. We have an attention layer where we have that is the source of 2 additional losses in addition to the focal loss that we use for multi-label classification. We also have a weak supervision loss, which is the Kullback-Leibler computed from the Kullback-Leibler divergence from the ground truth sorting signal. That is, we use the actual positions of the known sorting signals in the sequence as targets in the training. And we have a regularization loss where we use a discrete cosine transform, which allows for so it should be better than the Fourier transform when we have signals in the C terminus and the N terminus of the sequence. Yes, I mentioned for the multi-label classification, we use focal loss, and the classification head for sorting signal type was trained after the rest of the model was trained on subcellular localization categories. So the model trained on subcellular localization was frozen, and then we trained sorting signals. In principle, it would have been better to optimize both tasks simultaneously, but that proved rather difficult. Vineet can tell you more about that. So for the results, you can can I get a pointer here? Just a moment. Here. When we compare to DeepLoc1 and to the competing method YLoc+, which is also a multi-label subcellular location classifier, we see that we have that in almost all cases, the Prot-T5 version of DeepLoc2, that is the one that called slow at the website, is the best one. There's one exception here where the correlation coefficient for Golgi apparatus is a little bit better in ESM1B, the fast mode of on the website. But in general, Prot-T5 slow mode gives the best results. And the top row here shows the predicted number of labels per sequence. DeepLoc1 being a single label predictor always predicts one and only one category for each protein. YLoc has a tendency to over-predict multiple localization, so it predicts more than 1.5 locations per protein, where the actual number in the dataset is 1.27. And DeepLoc2, both modes are very close to the actual number. When we go to the independent dataset, we have two more methods we can compare to: FuelMLoc and LA-Prot-T5. These two were not included in the cross-validation testing because there's an overlap between their training set and our training set. And but for the HPA dataset, we could use them. The predicted number of labels, we can see again that while DeepLoc+ has a tendency to overpredict multiple localization, while DeepLoc2, especially the Prot5 version of DeepLoc2, gives a realistic number of locations per protein. LA-Prot5 actually underpredicts, in this case, multiple localization, and this number is lower than 1 because it in some cases LA-Prot5 predicts one of the categories that is missing from the HPA data. So again, the accuracy, which counts the occurrences of all labels being correct in a prediction, is better for LA-Prot5, but the other measures are better for DeepLoc2. The Jacquard score, the micro F1 and the macro F1. For the sorting signal prediction, we don't have very much to compare to, but we have here compared to specialized predictors of so we have a signal peptide prediction which is on par with the latest version of signal P. We have mitochondrial prediction also as good as the latest version of target P, while chloroplast and thylakoid transit peptides are not as good as target P. GPI anchors, again, not quite as good as net GPI. And this table on the right, it shows the Kullback-Leibler divergence between the attention and the known sorting signal. The lower, the better. So we can see results for DeepLoc1 and the 2 flavors of DeepLoc2. And note that for DeepLoc1, this is unsupervised training. DeepLoc1 was not told anything about sorting signals during training, while DeepLoc2 had supervised training of sorting signals. So what I want to pinpoint here is that some signals are very well defined, like the thylakoid, the chloroplast signals here, while we are really bad at pinpointing nuclear export signals. The web server looks like this. You can it runs a maximum of 500 sequences per submission, but academic users can download the program for free and run on their own computers. And note that you can select between high quality and high throughput model. The high quality, that is the Prot5, and the high throughput, the fast mode, that is the ESM1B model. And the output looks like this. There's the entire sequence is shown here with the relative sorting signal importance. This is the attention from the attention layer normalized so that the maximum value is 1. And you can see there are 2 predicted signals in this sequence, a signal peptide and a transmembrane domain. You don't get the precise positions of these signals, but you can correlate them to the over representation or the over attention in the attention plot. So the signal peptide is approximately here, and the transmembrane domain is approximately here. The note that the chosen localizations have a green background. The darker green, the more the probability is above its threshold. And here you can see that cell membrane is green, and lysosome vacuole is a lighter green, meaning that that value is closer to the threshold, so that's not so confident a prediction. Okay, that was actually it. I should thank the DeepLoc2 authors, Vineet, José, Alexandra, and Ole. And the web server URL is here, the DeepLoc2 paper, the preprint for graph part, the homology partitioning algorithm, my group homepage, and my email address is here. Thank you very much for listening.
Thank you very much, Henrik, for your talk. So, colleagues, please don't hesitate to ask questions. Now you can write the question in the chat, or you can unmute yourself now and ask by voice. So it's up to you what you prefer. Both ways, okay. So any questions, suggestions, comments, whatever?
Hi, can I ask a question? Thank you very much for the talk, and it was very interesting. Could you please give some details about how data was pre-processed before you feed it to the transformer structure?
So the transformer takes the transformer is trained on amino acid sequences. So it takes amino acid sequences and as represented with sparse encoding. So that's well, I don't know really what to say here. You just feed them to the transformer, and they encode it in as a high-dimensional vector.
With some, because sometimes some models works with multiplying alignment sequences, sometimes with some other way of pre-proceeding. So I guess it was some kind of one-hot encoding. Or...
Yes. Before it's fed to the transformer, it's actually one-hot encoded, and we don't use multiple alignments in this case. DeepLoc1 used multiple alignments, and that turns out to be a bottleneck for the web server. So we wanted to avoid this in DeepLoc2.
Okay, I see. And maybe thank you for the answer. If it's possible, one more question. Like,
course.
I mean, the location of protein is not permanent. I mean, sometimes a lot of transcription factors, for example, are located as well on DNA as well as in the cytoplasm. So what is the answer of your model in this case?
Well, if it answers correctly, it should give predictions of both nucleus and cytoplasm.
Oh, of course, yeah. Thank you.
You're welcome.
Colleagues, any other questions, suggestions, comments, whatever? So don't hesitate. You can write in the chat or ask by voice. You can unmute yourself. Henrik, may I ask maybe several questions? So first, this ongoing CAFA competition, we are trying to predict the gene ontology terms. One of the sub-ontologies is cellular localization. So it's quite similar to what it's doing, but as far as I understand, you have only something like about 10 kind of labels, but in gene ontology, I know thousands, yes, we have. So can you please comment on the possible relation?
Yes, we in our data set, when we extract it from SwissProt, we use the subcellular location comment in lines in SwissProt, and there's a hierarchy of terms there as well, the SL terms, and I don't know if there's a 100% correct mapping between the SL terms and the cellular component Go terms. It could be very nice to have such a translation table. I don't know whether that exists.
Okay, I see. So maybe a related question. So now on the top of the leaderboard of the CAFA competition, there is a team from Robert Hauchendorf laboratory. So they develop DeepGo Zero, and so it can also, so it's predicting these gene ontology terms. So I wonder, have you looked on there something papers and maybe try to compare with your approach, or no? It's maybe difficult, yes. So what's your...
Yeah, unfortunately, I'm not aware of that work, so I can't really comment.
Okay, okay, I see. Thank you very much. And so another question about this data preparation. So, do I understand correctly? So that you take this SwissProt data, and they have something like this labels, which you mentioned on the localization, like Golgi apparatus and so on and so forth. So, what's your opinion? What is the quality of these labels, and what are the experimental techniques which was used? Because I mean, it can be some experimental technique which is more more confined and some less. So what do you think about quality of these labels?
Yeah, the SwissProt doesn't tell us which actual method was used in each experiment. It gives the evidence code 269, which means experimental evidence. and then you would need to look up the reference for each annotation in order to arrive at what type of experimental evidence there was. So we have that would be an enormous task for 20,000 proteins, and not possible to automate. But the HPA data, where we know that that's they are assembled in a more stringent way using this confocal microscopy.
So haven't you compare this, for example, HPA labeling and UniProt labeling? Also, maybe there is some intersection between SwissProt and HPA, and then you can compare the labels, or it is impossible?
Yes, no, it is possible. We did look at this, and there are both agreements and disagreements between SwissProt and HPA. But in our HPA test set, we have only kept sequences that are dissimilar to the SwissProt training cross-validation set.
Okay, I see. Thank you very much. So, colleagues, any other questions, comments, suggestions? So maybe I'll ask another question. So now, so in this CAFA challenge, one of the big problems is again with these labels, because many proteins should have some labels, but experiments have not been done. So we can kind of think it's called something like open world, yes, because we think there should be labels, but just experiments haven't been done. And that is a big problem for the CAFA challenge, because training labels which we are working on are incomplete. So for your task, is there something similar? And if yes, then how do you overcome this problem?
Yes, definitely many proteins that should have subcellular location labels but don't have them. Proteins in SwissProt that simply lack the subcellular location command line. And we hope to be able to take advantage of those sequences anyway through the use of pre-trained language models, because the pre-trained language models have seen all proteins there are in UniProt, not just SwissProt, but also Tremble, and can recognize some patterns in these sequences without knowing the labels beforehand.
Okay, I see. Thank you very much. Colleagues, any other questions, comments, suggestions? Michaela, maybe you can ask some questions, and if you have some comments.
Yeah, I put a question in the chat because my daughter is excited to have a friend.
Sorry, I have not noticed.
But now he's quiet, so I can ask it. I was wondering how difficult it is to retrain this model with a more up-to-date version of SwissProt, or if you plan, to have update of the model. And also if you can see the code of the model in GitHub eventually.
Yes, it's not very complicated to retrain once you have the model architecture. Maybe I don't know if Vineet wants to comment on this. It was Vineet who was in charge of the actual training. Vineet, how much effort would it be to retrain DeepLoc on a new version of SwissProt if we wanted to do that?
So that should be fairly straightforward. I think the more so preparing the data set, I guess, it would be the main bottleneck here. But once we have that, the training should be it should be straightforward. So how much difference would there be in like the new version of SwissProt, or since this is like just like 2 years ago, right? So would there be like way more labels, or do you guys have any idea?
Well, no, I'm asking because the experience of this competition, if we got something, is that if you don't use the most up-to-date version of the train set, you are losing something. You most up-to-date version of SwissProt, you're losing something. So that was why I was thinking, it would be great if you could have a sort of pipeline that retrain the model every year, to keep it. So it's quite a burden. I mean, because you need to reevaluate everything all the time. But I think it's such a cool project that would be worth to have it up to date.
Okay, thank you very much. Any other questions, comments, suggestions, whatever? May I ask again a question? So, Henrik, can you please go to some previous slide where you have this with the domains, I think, the protein domains with the kind of motifs you have? Yeah, no, this one. No.
The
kind of
the data will this one.
No, a kind of motifs of the domains, I think. No, I think the next one. Yeah, this one.
Yeah.
So, so do I understand correctly that so there is some databases of the protein domains? Now it's in Interpro, but I forgot the name. So what is on your picture? So you have this kind of motif, as you say. So does it correspond to this some known databases, or it's something new?
So we see a certain correspondence to known motifs. We don't use Interpro or ProSite for labeling the known motifs. We use the data sets of signal P and target P, which are in turn derived from SwissProt, from annotation lines in SwissProt. So, for instance, the signal peptide here has been labeled in SwissProt with an FT signal line, and the transmembrane helix with an FT transmem line.
But I mean, these pictures, they appear from your approach, or you compare?
Yes.
Okay.
Yes. The picture is appears as output of the trained network.
So it's a kind of attention which points to this point, this kind of domains.
Yes.
Okay, I see. Thank you. Yeah, very interesting. And so you can find this transmembrane domain and this signaling peptides, for instance, yes. But do you mean you have such for all of the targets which you have for Golgi apparatus, lysosome, and or it's only for two?
Well, we have no annotated signals for Golgi apparatus because there are no described and annotated signals that signify Golgi retention. That is, the signals there are more unknown, less well known, and more subtle.
Okay, I see. Thank you very much. Okay. Okay, colleagues, any other questions, suggestions, comments, whatever? Don't hesitate to write in the chat or ask by voice. Okay, if no questions, then Henrik and the colleagues, thank you very much for the great presentation. It is very interesting, and we hope we can use it in CAF. But by the way, do you plan to kind of look on the CAFA data and maybe kind of participate, or?
Well, it would certainly be interesting, but I think for the time being, I wouldn't have the time. But maybe for CAFA 6.
Okay. So, okay, thank you very much again. So, colleagues, thank you very much, everybody, for participating. So maybe we can stop now and have a good evening.